So we do have another checkpoint that comes in at this point in time. We have the ATM nibrin protein, which comes in to check our DNA copies. And so we're going to come back to these that we copied. And all we're going to do is make sure that our copies match each other. And so these should be exactly the same. CCAGCT, CCAGCT, GGT, CGA, GGT, CGA. So these are correct. Then we'll come here and we'll check these. GAAC, GGA, TCT, GTAGA. Should match this one. GAA, CGG, ATC, TGTAGA. And they match. And the same for the others. CTT.
Chris.
Let's try that again. CTT, GCC, TAG, ACA, TCT. Should match. CTT, GCC, TAG, ACA, TCT. And they match. So our checkpoint did not find any errors, so we are good to go to our next step. It's also time to answer number 5 on your lab sheet before you move to our final process. You can pause here if you need to, so that way you can answer question 5. In the G2 phase, which is next, we are going to prepare the cell for division. And in order for that to happen, we have to create some new materials, synthesize some more organelles that don't normally exist inside the cell. And so what we're going to do is we're going to find our string and our paper clips here. And because we synthesized these 2 strands, we're going to use 4 of the string. We do have enough for 6 here. I went ahead and I did 2 of the sets of DNA for our lab purposes here. And so I will have enough just for that. Okay. For this particular step, what we're going to do is we're going to tie the string to our paper clip and make our spindle fibers. Now, spindle fibers are these very special organelles that come in only during cellular division because they are going to act kind of like a fishing line, and they're going to pull the DNA into separate directions or onto opposite sides of the cell, basically. Half of it's going to go in one direction, and the other half is going to go in the other direction. So that way the cell has 2 sets of DNA. So when it splits, each new cell will have its own copy of the DNA. And so these are the spindle fibers we're going to create that are going to be used in mitosis. So this is pretty much where we stop because we've prepared our cell. We have our DNA set up at its centromeres, ready to be separated. We have our spindle fibers ready to pull those DNA strands in opposite directions. So we are good to go. You have one last question on your lab sheet, question 6. You can go ahead and answer that. And then you are finished with this particular simulation. And your next simulation will actually take us through the steps of mitosis and continue and finalize our cell cycle.